So this week's lab is on macroevolution, evolution at or above the species level. So that means species, phyla, kingdoms, and everything in between there. Now, when we name groups, so think of Carl Linnaeus. Linnaeus came up with a classification system. He did not know about evolution, but he classified things based on how they look. So mammals were things that had hair and mammary glands, right? And he classified lots of organisms based on the traits they had. And so oak trees made acorns, and pine trees, made pine cones and had needly leaves, so on and so forth. He grouped things based on what they had and what they looked like. But now we use different ways of classifying organisms that are a little more complicated, that take evolution into account. And we use something known as cladistics. That's when we use shared traits that organisms have to help figure out their evolutionary history. And we use those techniques to build these trees that you might hear referred to as a cladogram. Cladogram is a word that you may see in your book. And also you might hear the term phylogenetic tree. A phylogeny, by definition, is the evolutionary history of an organism. And a phylogenetic tree is a tree that depicts that history. We are going to use cladistics to build a cladogram, which will represent the phylogeny. I know that's a lot of terminology there, but you're likely to hear both of those words. So just for our purposes, assume that we can use the word cladogram and phylogeny somewhat synonymously, and you'll be fine. Okay? All right, so here's an example. Let's say this tree represents the true phylogeny, the true evolutionary history of an organism. A clade, by definition, is an ancestor and all of its descendants. This is how we want to create groups like mammals and the domain bacteria, so on and so forth. We want it to represent all bacteria and any of the fossils that we find that led up to the living bacteria, okay? So a clade is an ancestor and all of its descendants. So everyone at the tip of this tree is alive in this cladogram, and down here represents fossils or hypothetical ancestors, okay? So here's a big clade. Here's a small clade, an ancestor and all of its descendants, right? So anywhere along this branch could be ancestors of the critter living at the tip. That means living now. Now, what happens is we sometimes find out that Linnaeus or ourselves later group things incorrectly because we looked at how we grouped them based on how they looked, and we get it a little bit wrong. So here's an example of where maybe we called this organism a snake. Over here are the true snakes. And so true snakes are here, but we called this one a snake when really it's a lizard. And we called them all snakes. And we realize when we study things using cladistics that one of those is not a snake and it needs to be removed from the snake group, okay? Likewise, we could have an issue like this where we name this group, but we leave somebody out. So it's, in this case, we don't have a common ancestor included in the clade. And in this case, we left somebody out. So this is like the birds. These are reptiles, and here's birds. And for years, we refused to acknowledge that birds were reptiles, but we're making changes in the right direction now. So we want to try to make sure that the groupings we have, like kingdoms, phylums, class, orders, families, etc., represent the actual evolutionary history of organisms. And so we're going to work on making trees today using cladistics. We're going to make evolutionary trees that reflect the history of those organisms. So here's an example of a cladogram using real organisms. So we have lampreys, sharks, ray-finned fish, lungfish, amphibians, and land vertebrates. So here's a cladogram showing the hypothesis of the phylogeny of these organisms, so how we think they're related. Now these are always hypotheses. Cladograms are always hypotheses, and they can change with new evidence. So for example, this entire clade we could call the vertebrates. Within the vertebrates, there's a clade here that includes everyone that has jaws. So down here was an ancestor with jaws, and it passed that trait off to all of its descendants. That's how we make these cladograms. We look at shared traits. So jaws are a homologous trait found in all of those organisms, and they share it because of a common ancestor. And within that clade, we have a clade that has bony skeletons, and within that clade, we have organisms that have lungs, for example, and so on and so forth. Okay, so there are clades nested within clades. Just like a kingdom has a lot of species in it, these clades have a lot of other clades within. Here's an example of, I said earlier, if we left out birds from reptiles, that would not be a clade. But if we include birds and reptiles and a common ancestor down here, so everyone in the blue circle is what we might call a reptile: lizards, turtles, crocodiles, and birds, right? So how would we make a cladogram like this? First of all, we would have to examine the organisms and look at their traits and figure out what traits they have that are unique and what traits they have that are shared. So unique traits we might call derived traits, traits that evolved recently that separate them from their relatives and their ancestors. Shared or ancestral traits are the traits that these organisms inherited and they shared together because of their common ancestry. So the first thing we need to do is we need to acknowledge here that group A is the outgroup. The outgroup is the group that is known to have diverged first. That doesn't mean it's extinct. It's not an ancestor to everyone on the tree. It's just the first group to have diverged from the common ancestor of everyone on the tree. And this is important because it helps you to figure out the order of the evolution of traits on the tree. Now this table down here we call a data matrix. And what we do is we code it with ones and zeros. So we have traits on here and ones representing present and 0 representing absent. So here is organism A, which we know is the outgroup, maybe from fossil evidence. And we say I is present. So who has eyes? Everyone has eyes. So A, B, C, D. Everyone has a 1 right there. If you add up this row, it'll add up to what? 1, 2, 3, 4. 4, right? Feet present. How many of them have feet? A, B, C have feet. That adds up to 3. Hat present. Only B has a hat. And lips present. C and D have those. So a lot of times you can add up these rows in this case and figure out which traits are ancestral. The traits that have the highest numbers are those shared by everybody on the tree. Those are ancestral. The traits that don't add up to very many are more derived traits that emerge later on the tree. And I want to show you the importance of knowing what the outgroup is. If I just showed you these organisms and told you to figure out who the first one on the tree was to emerge, you would likely say D, because it doesn't have feet and everyone else has feet, right? But if we know that the earliest members of this group had feet, that changes our interpretation, and that's why an outgroup is so important. So if we then take this information and we try to build a tree that shows these traits, and we try to make a parsimonious tree, meaning a tree that has as few changes in characters as possible. That's what we're trying to do here. We try to make a tree that displays as few changes as possible, meaning it's more likely that all of these critters have feet because they inherited them from an ancestor, rather than each group evolve feet independently of each other. Okay, so here's an example of a tree, and these hash marks represent where we could write what the traits were. So a trait shared by everyone on the tree might be feet or eyes, okay? Feet or eyes, right here. Everyone on the tree except D has feet, so feet are likely a trait shared by everyone's ancestors, okay? A trait that's derived for group B would be a hat. A hat could go here. It's not found in everybody else, but it is found here. Now let me show you. You could put hat down here and then put loss of hat right here, but that's an extra step. So we're trying to build a parsimonious tree, meaning a tree with as few steps as possible. It may not be correct. Maybe hats were shared by everyone's ancestor and then lost a couple times. But the first tree we make, we assume that things happen in the most simple way as possible, okay? All right, so what trait should go here? Well, I would put lips right here. Lips are a trait that unites groups C and D together as a clade. And then right here, what would separate C from D? That would be a derived trait, which is the loss of feet. Okay, so there's an example of one of these cladograms built using shared characteristics. So you can build these for living things and code them based on the traits that you know these groups have, and then you can build cladograms that show where these traits are found. And let me just emphasize once again, in this tree, sharks are the outgroup. That does not mean that we all evolved from sharks. It means that sharks was the first group to diverge from the common ancestor of everyone else on the tree. So always read these trees from the tips. That will remind you that everyone on the tip, actually, I said that wrong. Let me rephrase that. Look at the tips, that's everyone who's alive now, but look at the topology, meaning where the branches are, to let you know how things are related. For example, you might look at just the tip and think that rodents and rabbits are closely related to crocodiles, but that's not the case. If you go down the branch, you'll see that crocodiles and birds share a common ancestor right here, okay? And more common ancestors down here. Now, we have to be careful because when we build these trees, sometimes we might get confused by analogous traits. That could be analogous physical traits or analogous genetic traits. These are similar structures derived from different ancestors, so adaptations from unrelated organisms to the same environment. For example, the eyes of an octopus and the eyes of a human are and a lot of vertebrates, are very, very similar. So we might accidentally put mollusks as being closely related to chordates. But in reality, we can see that they're not. And that means that these eyes evolve separately because we don't see these same kind of eyes in worms or arthropods or echinoderms. So those are analogous eyeballs. Okay? So let's look at an example of this from your textbook. You see this organism here, a lot of us would call that a snake, and if I remember correctly, Linnaeus called this a snake. Okay? Looks kind of like a snake, right? It's a legless reptile. But if we examine it in great detail, let me exit out of here for a moment. If we examine it in great detail, we can see that this organism has traits. Let me go just straight to the finalized data matrix here. This organism, which is the new organism here. Here's the new organism. Here's snake. Here's lizards. And salamanders are our outgroup, okay? Lizards, unlike salamanders, have an amnion. Snakes have an amnion. That's a membrane that lets them lay eggs on land, okay? This new organism has an amnion, so it's probably related to snakes and lizards like you'd expect. It doesn't have legs, just like snakes, so we might think it's a snake. But it has external ear openings, which snakes do not have, but lizards do have. And this new organism does not have a hingeable jaw, but snakes do have that. One last thing is that it has movable eyelids, which snakes do not have. So based on all that, if you build a tree that shows the evolutionary relationships of this organism, you might realize that, hey, this is actually a lizard that has lost its legs. It is not a snake. Okay? That's an important thing to note here. Okay, so in today's lab, the first thing you're going to do is you're going to look at some imaginary organisms called caminacules, and you're going to fill out a data matrix for the living species. And then what you're going to do after you've put the living species on a cladogram, you're going to then put fossil organisms on the cladogram as well. So I will walk you through that real quickly here. I know this is a long pre-lab video, but it just has to be that way. All right. So you'll have a worksheet that looks something like this. So you have 1, 2, 3, 4 living caminacules. Okay, and you will put their traits on a data matrix, which is here. And I did one of the columns for you. So I put posterior appendages, which means appendages on the back side of these living things. Number 1 is our outgroup, and I coded that for you. So you can come up with other codes. Like, I wouldn't worry too much about spots, but look at their features like flippers and arms and eyes and things like that to try to figure out how they might be related, okay? So these are the living ones up here. These will be on the ends of the branches. So the way you'll put these on this cladogram, I already put the outgroup on here. You don't have to draw them or anything. You can put their number on the cladogram. The next thing you're going to do is look at these fossil examples, and these fossil examples have how old they are in millions of years next to them. So fossil 1 is 210 million years ago. So what you'll do is you'll realize that this cladogram starts at 210 million years ago, and so I put F1 here representing that's where fossil 1 goes. I did fossil 6 for you as well. Fossil 6 is only 50 million years ago. That means here's the present. All the living ones are at the ends of these branches. The present is up here. 50 million years ago would be about here, and fossil 6 actually goes right there. So I did those two for you to help get you started, because what you're going to do is follow the instructions. You're going to fill out this data matrix for just the living things, not the fossils. You're going to put the traits of those living things on the cladogram, like those examples in the video. You're going to put where the living ones go on the ends of these branches, and then you're going to put where the fossils go on these branches. So to help you understand how to do that, bear in mind, like I said right here, was about 50 million years ago. That means everywhere equal to about the beginning of this arrowhead, if we go to the right, everywhere up here is 50 million years ago, okay? Everywhere down here, even with the bottom, which is only right here really on this cladogram, is 210 million years ago. So if an organism is like 60 million years old, it would be down here somewhere on the timeline. So it could be here, it could be here, it could be here, or it could be here, okay? You'll have to figure out where it goes based on the traits that it has. The last part of the assignment, after you answer these questions, will be to do a cladogram based on actually it's molecular data. We're going to do it based on amino acid data, which is based on genetic data, right? Because amino acids are coded for by codons in organisms. So we're going to compare hemoglobin in several different organisms. We're going to look at the hemoglobin that they have. We're going to look at the order of the amino acids in that hemoglobin and compare it among organisms to see how closely related they are. And there's information here on how to do it. I'm going to run an example here for you really quickly. Okay? So you're going to go to phylogeny.fr. And it looks like this, and you just come down here to one click. You click on the one-click analysis. And on the worksheet, there's going to be a data matrix already made for you. I got that data from a website listed in the handout called Uniprot, which is a place where you get protein information. And if you ever want to play around with it, all of this is freely available. You can do this on your own for any organisms you want. It's pretty cool. But you paste the information here. So what this is hemoglobin data for humans, snakes, salmon, so on and so forth. The data that you'll be using is different. I'm using a different set of data here, but frogs and gorillas, okay? Then all you have to do is click submit. Now what it's going to do is it's going to look, these letters here represent amino acids. It's going to compare the amino acid sequences in all of these organisms, and it's going to try to put together the likely evolutionary history of this group based on how similar and how different their hemoglobin is. So I click submit. Oh, and of course I have an error. Hold on a second. Let me do that again. I pasted it incorrectly. Okay, so I paste the data in here. I had to use a slightly different data set. My other one was in the wrong format. And I hit submit. And then what it's going to do is it's going to compare the hemoglobin in the different organisms. And then what it'll do is after it processes, it's going to build a tree of how these organisms are likely related. It might take a minute. And sometimes the server's busy, so you might have to try it at different times. And it's going to spit out a tree. So I'm going to make this tree bigger so we can see it. Now this tree is a phylogram. It's similar to a cladogram. We'll just call it a cladogram for now. But the lengths of the branches represent how different the proteins were between the groups. Okay, so our outgroup here is this one down here, which in this case is a fish. And we can see that up here is a frog, and a platypus is here, and so on and so forth. You can see how these groups are related. So what you can then do is click on download as PNG, and you can you'll see something that looks like this, and you right click and save image as, and you can save the image. It'll actually look better than it looks here. You can save the image to your computer and paste it into your assignment. Okay, so that's how you'll do that. And I think you'll be able to figure that out pretty easily. All right, so you'll turn in your assignment to your instructor after you've completed the questions.